All right, welcome everyone to the Theoretical and Computational Biophysics group. I would say this is the best attended seminar of the year, actually, in 3 years, I would say. So it's a true pleasure for me to introduce our guest today, Professor Erik Lindahl, all the way from Stockholm. So Erik, by way of introduction, Erik got his master's degree in engineering physics from Lund University, and then his PhD in theoretical physics from the Royal Institute of Technology in Stockholm. And then after doing some postdocs with prominent groups around the world, in Groningen with Alan Mark, I believe.
It was, yes.
Yes, and then with Mark Levitt at Stanford, and then another postdoc in Institut Pasteur in France. He joins the faculty at the Stockholm universities, where he is currently a full professor of theoretical chemistry and also a professor in biophysics at Royal Institute of Technology in Stockholm. He's located at the famous Science for Life lab, which is engaged in really high-profile biochemistry and biophysics and structure research and many exciting subjects. Erik is truly one of the pioneers in the field. You know, from the very beginning, his early work was really pioneering how to look at lipids, how to connect their properties to macroscopic properties of membranes that we know, NMR, time relaxation, diffusion of lipids, and so on and so forth. He was also involved in implementation of a lot of algorithms, you know, fast simulations, electrostatic calculations, and so on and so forth. As many of you know, currently Erik leads the development of GROMACS, which is a very well-known software in our field. We even use it here, by the way, some of us. Thank you. And then actually what really impressed me a few years ago when I found out was that in addition to doing computational work, they started doing also experiments in their lab. So I was really impressed. You know, the early work of Erik on membrane extended into ion channels, naturally, protein-lipid interactions, and they started doing electrophysiology in their own lab, because I guess it was too much to wait for the collaborators to do it. Let's do it ourselves, right? Okay, so then they were also involved in development of algorithms in cryo-EM structure reconstruction. You might know about the program RELIAN, which is a famous part of any structure determination that you do. Actually, Erik's algorithm and contribution was essential for that program, which is now used by thousands of people around the world. So it's really a great pleasure. Oh, by the way, I forgot to mention this. So he has more than 65,000 citations, more than 130 papers, and his H index is 61. He's also heavily involved in supercomputing efforts in Europe, including policy making, how they should be used, how much investment should be done, and so on and so forth. So it's really a pleasure. Thank you very much for coming all the way.
It's great to be here, man. If you're embarrassed.
Thank you. Thank you. Look forward to it.
Thank you so much. Yeah, the great thing about hearing things like that is not about my CV, but when we remember where this field was like 30 years ago, right? That this is not my contributions, but it's great to see how much the field has evolved. Because it makes me remember that these tiny simulations, you know, 64 lipids, 10,000 atoms. The longest simulation in my entire thesis was 100 nanoseconds, and I was so proud of it. Yeah, you could do that in a lab today. So it's kind of amazing how much we've all learned over the last 25 years and how much computer has played a role here. There is a common theme of computers here, but there is also going to be some wet lab stuff. And we mix these things fairly heavily nowadays. We mostly just go in the direction we find fun. I realized I thought that I talked about this types of channels here recently, but I haven't, at least not the last 4 or 5 years. I'm going to do first a few slides about the reason we're working with this. This is Sir Robert Liston, Dr. Robert Liston at least. He was a famous surgeon, one of the most famous surgeons ever in all times, at least in the UK. The reason why he was famous, he could amputate a limb like a leg in 2.5 minutes or an arm in 30 seconds. And in those days, he used to be a good surgeon, was a fast surgeon, because while you were amputating that leg, the patient was lying on the table screaming. Wow. He has also said that at some point he removed an entire limb in 28 seconds, but unfortunately he managed to somehow cut the wrong way so that he cut off his assistant's finger. The assistant died of sepsis. The patient died due to the cut or something, and another spectator in the audience died from shock. So this is likely the only surgery with a 300% mortality rate. But jokes aside, that it's easy to, when I mentioned to him over dinner that many years ago, 25 years ago, when I was thinking about what I want to do with life, I could either decide to go into physics or medicine. I could be interested in almost anything in medicine. There was only one thing that I found completely boring and uninteresting, and that would be the anesthesiologist, just standing and making sure the patient doesn't die. And yet there is so much of our modern advance in medicine that is due to the fact that we can sedate patients. It doesn't take 28 seconds to amputate anymore, but you can actually sedate a patient, and if you're lucky, you revive them again. So this is a classical event, Morton Auditorium in mid-October 1846. Actually, not this one. The first one was 2 weeks earlier, but we don't even know what the patient was, but they were able to extract the tooth from the patient without pain. And then when Henry Bigelow heard about this, he arranged this surgery of removing a tumor from somebody's neck or something. And the patient actually survived. And it's classic photo of the first ever surgery performed under anesthesia. And all this is trial and error. Modern anesthesia is almost 150 years of trial and error. Until recently, we had absolutely no idea what is it that made you go unconscious. The one thing that people learned, and this is 100-year-old results, there's a classical Mayer-Overton correlation, and this correlation is so good. So if a student comes up with something like this from a lab, you would assume that they cheated. So that over 5 orders of magnitude, the more hydrophobic a compound is, the better it is as an anesthetic. And already 100 years ago, we knew that membranes consisted of lipids or something. I mean, obvious that the anesthetics must somehow go into the membranes, influence the membranes in whatever way. We had no molecular models at the time, or they did. And that was the entire hypothesis of how anesthesia should work. And this was a very clear alternative until some 15 years ago. And then gradually, though, there were a few modern anesthetics that got developed that started to deviate from this, some of the most efficient ones, and they were fluorinated or brominated compounds in particular. And I'll come back to that in a second, but the reason why this is super interesting to us is that over the last 3-4 decades, we've understood that what actually happens in the nerve signals is that you have a neuron here, where you have a voltage-gated channels, push a nerve signal, and that leads to release of neurotransmitters from vesicles. There are probably 5 Nobel Prizes there. They diffuse over some tens of micrometers here, and then you have ligand-gated ion channels sitting here on the receiving side, and if the right receptor hits them, they will open and create an impulse here. And some 30 years ago, there started to be more and more groups arguing that these anesthetics would actually target these channels instead. And there was a ton of debate, and as always in science, some of these things that look like horrible fights, they're interesting because the fights means that we're actually starting to make progress. Without going into too much detail about those fights, today we know that anesthetics in particular, they target these channels. These are amazing channels. So when I first got interested in this problem, that I was a student, a postdoc at the time at Stanford, and there's this crazy anesthesiologist that kept showing up, wanted help modeling this. A part of that, we have absolutely no structure. There are no homologs of no structure. We know nothing. There might be some helices in the membrane, and it's 5 times 350 amino acids long. And that's where you just sigh and tell them to go away. But for some reason I didn't. The amazing thing is that they're like Dr. Jekyll and Mr. Hyde. They can either be anions or cations, depending, but if there are 2-3 mutations you need to make to turn an anion channel into a cation at once. They're also regulated by these neurotransmitters. They can be acetylcholine, that's one of the most common receptors, but they can also be glycine or GABA, small amino acids, basically. Very small, simple compounds that bind to the exterior here. And then depending on the type of channel, that we can either steer this by preventing the excitation of the new nerve signal, or we can amplify the effect where we actually inhibit it. So that I'm not going to go into all the biology here, but without even having given you any details, there is more than one way to skin this cat. The cool thing is also you can't really open the channels in any way just by all this modulating things with anesthetics. They can only act as grease or a lubricant or prevent it. But somehow these modulating sites bind in a different place than the normal neurotransmitters. And it's great to give this seminar at UIUC because I don't, normally in many cases, it's really difficult to study eukaryotic channels, right? And for this channel that so many of us, including the voltage-gated ones before them, the reason we have good models for this is that Erik Jacobsen, I heard passed away, that he actually sat on his chamber with a student at the time, and they were randomly dug into different channels and started going through these databases until they found a hit in a bacteria. And that's a bit strange because bacteria are known for many things, but not their well-developed nervous systems. So we don't really know why these channels exist in bacteria. They might be pH regulated or something, but exactly what they do, we don't care about. But they're amazing motor systems because this suddenly meant that we could determine structures. And Erik he did this not just for one, but like he did for the first voltage-gated channels, the ligand-gated channels, it had the cyclic nucleotide-gated channels. And it's a bit sad that he didn't get enough recognition for this because we wouldn't have this field without him. As a result of those collaborations, and this is my last intro slide, a few years later, we managed to build these models because we had this first prokaryotic structures, and we were super happy because I was finally predicting binding just between the subunits here, and I felt that finally we were able to show something cool with molecular simulations before any of the experimentalists proved where the structure was. And then 2 months later, our close dear experimental collaborators published a structure where it had a beautiful binding site right inside each subunit instead of between them. So we had a 50-50 shot and we got it wrong. And at that point, I said that I'm going to kick out the computers and start an experimental lab. And yet, I have no problem being wrong. I've been wrong lots of times in life, right? But this was one of these cases. I was so sure that I could have eaten my left shoe. And then we were wrong anyway. And that worries me more because being wrong if I wasn't certain is one thing, but being wrong about something that I would have, again, I would never have guessed that could be a third. That worried me more. And that was one of the reasons why we started these experimental studies, because in hindsight, there were a ton of things we did wrong here. That we were studying one mutant where we tried to modify these channels to behave more like the human channel, while the experimental X-ray structures at the time, they were conducted on wild type, so there were like half a dozen of minor things that differed. So we weren't really sure what the difference was. And I think our early success here is that once we actually started with comparing apples to apples, it turned out that there are two binding sites. And at the time, we proposed that one of them was the on button, the activating, the potentiating site, and the other one was the off button. In hindsight, that was wrong too, because both of these are on buttons, while the off button is pretty much by plugging something in the middle of the hole. And this is just in the transmembrane domain. So there are already at least 3 sites there. And this site that we thought was simple in the extracellular domain, well, we probably know there are at least half a dozen sites in the extracellular domains that binds benzodiazepines or anesthetics or alcohol or you name it. And this makes these channels amazing, but also very, very complicated to understand. So we kept studying this, in particular with electrophysiology over the last few years, to really map out where we have these binding sites. So electrophysiology is very simple. I have a super skilled coworker, Rebecca Howard, that does all this stuff in the lab, but I spent a week with Pei Tang in Pittsburgh, and I've actually done it myself. You have these small frog eggs, they're roughly 1 millimeter in diameter, and then you just inject some DNA straight into the nucleus, and you put it in the fridge, which they call incubator in the lab. And then you attach, wait 3 days, and at that point, 99% of the membrane protein is hard channel, and then we can measure the current through them by putting these electrodes that you see here under the microscope. It's not hard. And the cool thing here is that then we can see different effects here. So this is an acetylcholine receptor. And in this case, when we add acetylcholine, you see that the signal is dampened, while the GABA receptor here, when we add the general anesthetic, you see that we get a slight increase. So they respond in opposite ways to the anesthetic. This anesthetic dampens, inhibits the acetylcholine receptor, but it increases, potentiates the GABA receptor.
The same anesthetic.
The same anesthetic. And this is a recurring theme of these channels, right? You don't even know which direction they're going to point in, which is super cool. not only that, I'll come back to that. So that we spent a lot of time trying to understand these sites at first. A few years ago, we were able to work together with Mark Delavry and Haira, who had determined the first X-ray structures. And this is kind of cool, but it's the first paper collaboration with Mark. There was some computation in this paper, and that's done by Mark's postdoc, while our lab's only contribution to this was the electrophysiology. So it's kind of reversed. Normally, Mark would do the experiments. But this is from the labs that had previously identified the sites inside each subunit. And suddenly we were able to show binding both in the plug, inside each subunit, and between the subunits, depending on the specific mutants we made. So now we can start making very simple single site mutants that create binding sites in different spots here. The other thing that we can actually show that we have a very clear size effect here, that depending on how large this pore here is, we can get it to match either propofol or bromophore, which are basically different anesthetics. So there's some sort of selective fit effect here. If the hole is large enough for a particular molecule to bind there, the molecule won't force the channel to open or close, but if the molecule fits just perfectly, it will bind in the pore, and then the pore is going to be so well packed that it will no longer open. Now, what we then did, or my students here, we started looking, really understanding what the difference between this at the time that we thought we had an on and off button. And we did a ton of simulations here. And I think we increasingly use simulations as a Gedanken experiment, frequently very simple simulations, until we have some sort of hypothesis that we think this is the way it works. And then we go into the lab, and we're probably right in 2 cases out of 3. But 1 time out of 3, the experiments say that we were horribly wrong. Which is humbling, because I think we love the simulations here and they're great, but remember that they're just models. So another thing that we found is that we can influence the accessibility to these sites. Again, just by, in the wild type of these cells, we have a methionine here, and just by changing that methionine to, for instance, a tryptophan, do you see again here, PFL means propofol, and this H+ just means that we're changing the pH. So in the wild type here, propofol, that anesthetic, would have an inhibitory effect. I think we talk about 1 single residue out of 350 in each subunit would change. And now it's just a potentially the opposite effect. If you think that's complicated, it gets worse. So this is again the methionine. So we start out here, in the wild type, the more we add, the more we inhibit it. In the mutant, I add a little bit of propofol, it goes up, it goes up, it goes up, and then I add more, it starts going down. So you have a constant, it's not just that the effect depends on the sign depends on the concentration. From here, from 100 to 300 micromolar propofol, they swap the sign. And this, if you think a little bit about it, imagine these channels are also sensitive to, for instance, alcohol. What happens, we've seen this effect in many cases. What happens if you take 1 glass of alcohol, right? It's a social drug. You get a little bit excited and you're happy to chat. After the 10th glass of alcohol, you instead get drowsy and tired. And there are likely similar effects here. And I wish I could say certain what happens, but our idea is really that you have these, you start by binding these very high affinity sites that are the ones that open the channel, and you open the channel more and more. But what happens when you've saturated all those 5 sites? You start binding there too. And when you start binding there too, that's when this works. But it's cool because it completely violates all the normal binding simply because we have more than 1 binding site. And that's the one take-home message. So, in the interest of time, but we ended up having a bunch of different models here. That what we still can't really explain is that this Meyer orbital, why do all these compounds have to be so hydrophobic? So there is something deep here. And if you look at them, an obvious hypothesis is that they somehow had to enter through the membrane, right? Yeah. And we still believe that. And there are these binding sites are likely more accessible from the membrane. And then here too, we have some sort of selective fit. And what made us, I think what made us crack that was a mistake, or not so much a mistake. So Stephanie Heuser was an outstanding PhD student. She kept coming back and said that there is something wrong with the equipment. I think she's a super skilled student. There's something strange because that these signals. sometime she got these memory effects that normally, if you add a compound and then you wash it out, you would expect things to be back to normal. That's how our normal channels behave. And in this case, we applied the compound here and then we wash it out, but there's a memory effect. And this memory effect can last for up to 30 minutes. So we really tried to wash away all the compound, but the channel somehow still has a memory. And we were convinced that there were impurities in our cabling systems or something, but no, that we tried swapping everything twice, and this effect still remains. So when we finally gave up and couldn't come up with any other hypothesis, of course, we somehow have these compounds must have partitioned into the membrane, and then we must somehow have changed the accessibility to these sites and made some of these sites so deep that even when we try to wash it out, these compounds stay bound to the channel. Sorry, this would be the wild type. But in this case, with the proline mutant, they bound so deep that even when you wash out, some of these compounds will still remain, and eventually they will partition back. And this could, well, we think that this, of course, where this hydrophobicity comes from, because that this will only work if they first enter spontaneously in the membrane and then always bind from the membrane, but never go through the pore. The cool thing here is that we were able to confirm this with simulations. So we can really show, we managed to show the accessibility, we managed to show how these compounds diffuse in and out, that Marie Luxel did, another outstanding student who didn't have a computational background at all, biotechnologist who came to the team. And the fun part, in terms of simplicity, these simulations are almost as simple as the ones I did in my thesis. They're much longer, of course, but there are no fancy non-equilibrium statistical mechanics or anything. Very plain, simple models. But the idea is, of course, they're models that are testing very specific experimental hypotheses and that we then can validate against experiments, because this would have been worthless without the experimental confirmations. And we studied this back and forth. I promised myself that we're never going to do anything more complicated than electrophysiology. Unfortunately, I'm not very good at holding my promises. So there was a class in Stockholm on membrane proteins, and we go through this, and I gave roughly this introduction to the class. And then we had this young student coming up afterwards and say, this sounds really interesting. She would like to do a thesis project on this, but the one thing that she's good at is biochemistry and purifying proteins. So do you have any use for that? No. But I could try to determine a cryo structure of the channels. And at the time, we just got to start in this I would only do computational cryo because we had a facility for this in Stockholm. But worst case, pretty good. So 2 years later, we had structures of both the open and closed states of the channels determined on our lab. And the cool thing that we're starting to see here, again, there have been prior X-ray structures. There's an X-ray is beautiful. You have a fantastic resolution. And I could never compete with the structural biology groups. But for us, one of the advantages of doing a simple experiment, I shouldn't say simple, but I could not do this experiment, but thank God Urska and Rebecca. I was interested in having structures that were pH dependent. And by doing this ourselves, we could actually determine structures under exactly the same condition with the only differentiating factor was the pH. And we were, to tell the truth, a bit disappointed because we had so hoped that normally the channel would be closed at pH 7, but open at pH 3, and it's not really substantially open at pH 3. And there was a reason it would have been that. The one thing we did show, though, is that we have significantly low resolution in this closed states, while the low pH, the one that was supposed to be open, that isn't open here, is much higher resolution. So we started to have these ideas that the resting state would really be characterized by a much broader distributions by these channels being fairly flexible, would also be the reason why, because we've averaged this over many different particles. While the open structures would be much more shorted, much more specific, and once you've been open for a while, you would then head over to the desensitized state. The last part is the only thing that we were right about. I'll keep you in suspense there over a reason why I don't think it's bad that it's not open. So armed with that, we had both through X-ray, cryo, but a few other structures. So we do have structures of these channels, at least at the end states, open and closed. We have no idea what the quality of those structures are, but having used Markov state models and folding at home and everything, I had another smart student, Catherine Burke, who's a super skilled physicist. and she knows the theory about all the Markov state models and various descriptors much better than I do. So she came up with this idea that at the time we had like 50 different experimental structures. So armed with that, we can just determine some sort of simple principal component space and interpolate between these open and closed states. And this is just the first rough guess, but we're fairly happy with the guess because there are also some intermediate structures and they happen to fall exactly on the path that we had predicted. And the idea then is that if we have these roughly 50 or 100 seeds, we have no idea about the quality of them, but if we then start long simulations from each of these seeds, and I think she collected roughly ballpark of 100 microseconds, no, there's 10 microseconds of data for 100 points, so a millisecond or so of molecular simulation time, but spread over 100 different simulations. Then, well, just by doing this sampling, then we can try to afterwards group this and try to determine a Markov state model. So really, can we somehow define discrete states and come up with a high-level model for how we're moving, and then ideally correlate this between open and closed states? And our idea here is that, under normal resting conditions, high pH, then if this really works, we should have a free energy landscape that says that the closed state is lowest, while the open state would have a high free energy, right? And when you change the pH, they should trade places. Mm-hmm. You agree? Mm-hmm. Good, you're as stupid as I am. It made sense. It made so much sense. And it's wrong. It's wrong for a good reason. I was disappointed when we first saw these curves, and in hindsight, they were so right, and I was so wrong. So Catherine, this is time-independent so-called TCA space, but think of this as some sort of principal component space of the two lowest degrees of freedom, just to visualize this. So what this is showing you is a rough free energy. So deprotonated, this is at high pH, where we would expect the channel to be closed. And all these structures here are various closed or locally closed X-ray structures. So we actually do predict that the lowest free energy should be in the states that's closed. While in the state that corresponds to the open, we have a relatively high free energy. Good, that makes sense. Then we protonate this, take a jump to low pH, and then we would expect them to trade places. And sure, we have a local minimum here at least, right? And that made me slightly happy. And we kept asking Catherine, can't you collect more data? Collect enough data until the results show us what we want to show, and then we hit the stop button. Well, there's more than one simulation that will let us be conducted that way. But let's not joke about it. It's fairly clear that this is still the lowest free energy. The part that we like, though, but this might be our force fields that are screwed up, or the models, or the sampling. So just at least getting some sort of result, let's go after a relative effect instead, because we've done so many experiments, and the advantage of doing these experiments with ourselves, we can then go after mutants, and we can tailor-make these mutants. And in particular, take a mutant that, for instance, makes the force slightly more hydrophilic, and that should make the channel open more. And we roughly do that. So in this case, we get a slightly lower free energy already in the deprotonated state, but in particular in the protonated state, the local minimum here is now larger. So we're reasonably happy with that. It's not a stellar result, but it's definitely we're predicting that the shift happens in the right direction. Mm-hmm. And then we submitted this to eLife, and then we got the reviewer coming back, the one we didn't want, and well, you might just have been lucky because it's kind of 50-50, right? So that you also need to do one more mutant. And then we had this other mutant that basically made the transitions much harder to happen, that slowed down the kinetics exactly where, and we were pretty worried here for 3 months. But eventually we managed to show this here too, that we get a much higher barrier here, which I think is awesome. But now here's the point. That the reason I was wrong, these results are much better than I expected, because the open GLIC in particular, we don't know exactly what the conductance is, but we know from virtually all of these channels, they do not have a 100% open probability even at activating conditions. So what happens is that normally at resting conditions, all the channels are closed. When you activate it and see a signal, that signal does not come from 100.0% of the channels. That comes from, in this case, probably 5, maybe 8% of the GLIC, a very small part of them. But 95% of the channels are still going to be closed. And if you have more channels closed than open, that means that the free energy should still be lower. So that this is mostly qualitative, but the whole idea is the difference we're seeing here. I think in our case, this corresponds to 8% probability, which is, to tell the truth, we were probably just lucky. Either anything within a factor 2 would have be happier. But this is so funny to see because my first impression here was a bit of disappointment. And in hindsight, had this been lower, we would have had a horrible problem because it would have been incompatible with experiments. So let's see. The other thing that we can then do with Markov state models, now we're not looking at free energies, but with Markov state models, we can actually cluster these and roughly, we can pick any number of states, but we managed to do roughly 5 large clusters. You can also make it, we can also try to go for only an open versus a closed state. So in this case, this would be the closed and locally closed, and this would be the open states. And to make a long story short here, everything matters. We get beautiful decreasing open probability, decreasing probabilities of observing things in the deprotonated states for the ones that would be open, and similarly increasing probabilities of observing them in the protonated states. But what the Markov states models also allow us to do is that it's in principle the same data that's in the free energy plots, but we can also determine kinetics. So between all these open and closed states, we can determine what is the expected transition rates. So we're talking about, well, half to 1 microsecond time constant for moving from open to closed. And I think all of those of us who have done simulations of this, we know that it's really hard to stabilize them in the open state. They so prefer to close. They are really good at closing. And while when we move to so-called activating conditions, they can open, but it takes a ballpark of 10 to 15 microseconds for a single event to open, while they still close faster. So this is a fairly simple channel. The one thing that happens for real channels, we're not entirely sure what happens to GLIC, that real channels, when they activate them, in this case, it's just pH-gated, but real channels that bind, well, eukaryotic channels that bind the ligand binding itself, it's low pre-energy, right? So it will take a while for that ligand to dissociate. And while the ligand is bound, all the windows are going to be open straight into your cell. You don't want that to happen. So for some reason, all these channels are created spontaneously. They open, but then they spontaneously desensitize without releasing the ligand. And what we think we are seeing here is that up here in the landscape, we appear to have a state where the lower, and it's not entirely easy to see, but we appear to have a state where we're losing the hydration in the form. So we might actually start to see, in particular for these cells, some sort of state that might be desensitized. We didn't dare to stick out, well, we described the dehydration, but we didn't dare to stick out our neck in the paper and actually say that it's a desensitized state, because we have no idea whether GLIC desensitizes. The other thing you can do with models like this, we can study asymmetry, but in the interest of time, I might have
to.
Yes. Yes.
What did you say? We don't have any idea as to whether GLIC desensitizes?
I think it desensitizes.
This is a functional definition.
Yes.
If you lower the pH of the channels open.
Yeah, but if it asks you, the mechanism of pH activation is not entirely the same as ligand activation, right? So the pH, the second you decrease the pH, you're not going to have any remnant effect on the pH that it stays deprotonated. So that the difference of a pH jump versus actual ligand binding is not entirely the same. That's why I need to be a little bit careful. But there's definitely the functional effect is there. So, yeah. We can see asymmetry to that, and this goes back to the we can see asymmetry to, and this really goes back to what I said, that the closed states are much more asymmetric, while the open state is again, this to be fairly short-lived and fairly symmetric and nice, and then we somehow drop into this desensitized state. Partly, one reason why it's partly fuzzy there, but for GLIC, it's not entirely easy to structurally decide whether a state is desensitized or not. For an acetylcholine receptor or a GABA receptor, that would be a structure. If we see that it has the agonist bound, right? But the pore is in a non-conducting conformation. For GLIC, we don't really see the proton. So we're starting to be fairly happy here that we're roughly understanding these relatively free energies, and in particular that there are relatively small changes in the relative free energies here. And then also that in some cases we're influencing the barrier here a bit. But in the interest of time, I'm going to need to continue. We're still working a lot with the prokaryotic channels, and I'm going to come back to that. But a few years ago, we started to work a little bit with Ryan Hedge's group, and in particular, Yuquan from our team started to work with them. And Ryan is doing far more advanced structures than we are, in particular going after heteromeric receptors. So this is an nicotinic acetylcholine receptor. And Yuquan has been able to show with simulation here too, that in this case, we also have binding, but in this case, you have nicotine binding up in the extracellular domain. And here too, we can show with simulation how binding either of nicotine or one of these nicotine mimetic drugs bind in these two specific sites, because again, this is no longer homopentameric. And here too, simulations appear to show how the binding of ligands causes these subunits to stabilize and basically turn in and bind around the ligand. We were also able to use simulations here to model what actually happened in the intracellular domain. So for these, so in this channel, there is some sort of strange density down here that we eventually resolved to be a lipid, mostly with MD simulations, because we can actually see a lipid. If we put lipids close here, the lipids slides in and binds here. So in this particular channel, while it's open here, the ion conducting pore does not continue straight through. It's actually blocked down here on the intracellular side. So instead, you have an ion conducting pathway here that kind of looks like a small pentapus, so that the ion pore here goes through here, but then it goes out between these intracellular domain helices.
This is pretty far from the membrane, right?
This is so the transmembrane domain. Oh, sorry. This is the top of the transmembrane domain, and that is the bottom of the transmembrane domain. So that stretches far out into the air. And this is stuff that we're about to submit now, that Yuquan has continued work with this. So about a few months later, Ryan's group were able to determine separate structures of the acetylcholine receptor in a resting or even inhibited state, so definitely close, an activating state, and a desensitized state. And this really is, for us, this was one of the first time we have all these three different states. And I know that this might not be a revolutionary study, but we wanted to, what frequently happens in all these structures, people have these extreme fights about whether is this really a conducting state, is an open state, is a desensitized state. So Yuquan decided to try to assign all these states based on molecular simulations and computational electrophysiology. So first, this was one of the first examples when we actually started to use massive coarse grain simulations in the team. I've been a little bit skeptic to that. It's awesome. Anything that has to do with sampling lipid distributions around membrane proteins, I have no idea why it works, but it works. But then, of course, after the sampling, you map it back to atomistic resolution, and then we can calculate free energies. And this, in our case, we use a technique called adaptive waste histograms, but you could probably use plain umbrella sampling too. The key thing here is that the difference in the pore radius between these cells is relatively small. There are, yes, some of them, the activating state might be slightly more open here, but they're not completely different. We talk about the difference in radius of less than 1 angstrom. And the lowest parts here elsewhere are suddenly fairly compressed. So it's not obvious.
So these are atomistic radii.
Those are atomistic radii after back mapping.
Right.
Sorry, those radii are even, these are even from the X-ray structures. Sorry, structures.
I see.
So what we do with computational electrophysiology is a technique that Carsten Kuster has developed in NPI is that you just copy the system, and then you can have, by having different concentrations of ions in this water bath and that water bath, and then keep adding or removing ions, we can just calculate the effective conductance here depending on the potential you have. And this works. It works remarkably well. And then you repeat this many times for many different charges. And then you end up with conductances here, both outwards and inward conductances that are slightly different. And they're talking, let's see if I run it, this is roughly 200 plus picosiemens, and it's roughly 44 or so. Now, what Yuquan was also able to do is actually calculating the free energy of various ions in all these three pores. So first, in the inhibiting state, sodium ions, we have a astronomically high energy. That's good. This channel is not conducting anything. And remember, we're not talking about extreme, it's less than 1 angstrom difference in the pore radii. In the activating states, for all these three different ions, it's mostly it's likely this is a channel that's binding calcium, but it's mostly calcium and sodium around here. But both sodium and calcium, we have low free energy pairs at most 2 kcal or so, which is fairly impressive, giving a 1 angstrom difference in radius. But then in the desensitized states, this jumps up to 5, 6, almost 10 kcal. So we appear to at least qualitatively map out the differences between all these states, and it's really only the activating state that is able to conduct ions, while the inhibited one is definitely non-conducting, and even the desensitized one has 2-3 kcal might not sound like an extreme difference, right? But compared to KT, 1.2 kcal would mean roughly 1 order of magnitude lower conductance. So 2-3 kcal would mean 100-fold. The other neat thing, let's see here. Yes, this is another one of those things we at first thought was almost an artifact. What Juan showed that both the inhibiting state and the desensitized state behave similarly in terms of the membrane thickness. But we repeatedly got this strange effect that in the activating state, the membrane has somehow thinned out. And it has thinned out by like 3-4 angstrom all around the protein. And the more we see of this work, we can't know that it's short because this is only a computational result for now. We don't see the lipids in X-ray. But this might very well be some sort of springboard that we're putting tension in the membrane here. Remember what I said, that you have one narrow state, and it has to be advantageous in the sense that you want to go there, but you do not want to stay there. So this might actually be an effect that the membrane tension here helps make this state somewhat unfavorable. So that yes, with the ligand bound, we will stay there for a while, but the membrane dislikes this so much that we're rapidly going to push it into another state where it relaxes a bit more, which is then non-conductive. So that would then help create this effect that you open translucent to let through a few thousand ions, but then boom, then you slam the door shut, even though you're still at the agonist bound.
But then you need to be lipid dependent, right?
They are lipid dependent. These channels are extremely lipid dependent.
Okay.
And you beat me to it. So we actually see state-specific cholesterol binding here too. Like this is not the same thing as showing that cholesterol activates. And so remember, here we are starting from the different structures, but we're seeing cholesterol molecules intercalating deeply between these structures, and they're doing it in slightly different ways. In particular, the desensitized state here appears to go further in. This is probably, I wouldn't say anecdotal, we definitely see these results, but we certainly haven't proven that it's a lipid effective. The other cool thing though is that,
If you go to more, you can hide that floating panel. It's a hide floating meaning control.
Hide floating meaning control.
There you go.
The other cool thing is that we can actually show something simulation that we did not see in the cardio, which is fun. So normally, at least for us, I frequently feel that we do post-diction in the simulations, right? The one sad thing about these structures is that right now they did not really resolve the binding sites that well. And you saw some sort of scattered density in all of them. In our case, in the simulations, in particular in the activating state, we see 2 beautiful binding sites, and that's exactly where we would have predicted them to be in the two subunit interfaces. And then we see this, in this case, a positive allosteric modulator drop. And we don't see that in the inhibiting state. And again, even if we're adding this compound to the membrane, it can't really bind in the inhibiting state. And in the desensitized state, we have some sort of density, but not for as much. So I think we're not quite there yet, but we are getting close to the point where we can actually use simulations to predict some parts of drug binding and showing how these states are specific. So roughly what we've been able to show is really that we have the inhibited states, and we've been able to show that within the simulation, adding the agonist, we actually get them to be stable in an open state that is conductive. This open state can go over to a desensitized state, and we see that the desensitized state in our simulations is definitely not conducting. And in both of these cases, I didn't talk that much about it. So if we can add one of these positive allosteric modulators, and then we definitely force the entire protein to stay open, and then it won't desensitize. I'm going to speed up a little bit here because part of the title that I picked here was lipids and another experimental technique, and I'm going to spend a few minutes on that today. So what we would all love to do is study these channels under conditions that are as realistic as possible, right? And at some point, I feel that X-ray is full of complications, and cryo-EM is such an amazing structure. But if you think about what you really would like to study in biology or something, is this should be a movie of flying birds. And if this is a simulation, you might get the flight, but you would only see the wing moving 1 millimeter here. That technically it's right, but it doesn't really teach you a whole lot of bird flight. The problem, oh yes, there we have it, that's what we would all like to have. And then we approximate this with X-ray cross-tomography. I'm not sure. Some of you might. I'm a structural biologist nowadays, so I'm allowed to make that joke. The problem is that cryo-EM, the first part of cryo-EM is not a whole lot higher temperatures. Cryo-EM, sure, we don't have a crystal in cryo-EM, so we don't have packed turkeys, but there are definitely frozen turkeys in cryo-EM too, and that's the problem. It's that they're not flying birds. In theory, we would like to use NMR. NMR is hopeless for a molecule that starts, at least for me. Some smart people in the audience did it more. And I think this is an old joke that Helmut Grubel used to make, that if you instead do bioinformatics or something, that you it's some sort of model like this. But as happy we were as these cryo-EM structures and everything, we somehow wanted to probe, does this actually correspond more to what we would have in real life, in some sort of more natural environment? Does this, I can't do anything with NMR. The idea would be to use small angle neutron scattering. So small angle neutron scattering is one of these very primitive techniques that I would argue is the technique that's roughly where cryo-EM was prior to the resolution revolution. Nobody would use it for anything in real life, but in theory, you can determine small angle neutron scattering, at least get the rough shape of a molecule, and you have to believe it's very noisy. But the cool thing that if it works, you're studying a sample in solution at room temperature, and you could even do it with lipid or a nano droplet or something, a nanodisk. The reason why we started doing this is partly financial. They're building this gigantic facility in the south of Sweden, which is a European Union infrastructure, which is a very powerful neutron beam. And that means suddenly that the Swedish Research Council had lots of new funding for projects in this area, and suddenly we saw the opportunity. There's funding that we can apply to, and just in theory, this might be a technique that could help us resolve these challenges. But of course, today the neutron sources are not very powerful and anything, but whatever, we had to go, and that's when we hired this Marie Luxel student. She didn't know what she was getting into. And this was just supposed to be, we figured that we're just going to model it, we're going to run some MD simulations and prove that at least in theory, we can calculate a neutron spectrum. Apparently, the dean in charge of that graduate school had a different idea, because he kept arguing that, well, you're going to need to do experiments or something. So Marie started having to purify this protein and actually try to do experiments on it together with a great group in Denmark, Lisa Ahlet. So this was just supposed to be a small one-off project, but it probably took her 2 years to learn everything. The cool thing you can do with neutrons is that you deuterate the lipids. They are filthy expensive to buy, so every experiment costs like $5,000. So you have deuterated lipids, and that means that you can make these nanodiscs around the membrane disappear, so that you're having a solution where you're only seeing the protein, but we're not seeing the lipids around it. And then the idea is that, can we now measure this under different conditions? That would have been a simpler experiment had it not been for the fact that these channels start to aggregate. Then you spend another year trying to come up with different ways to prevent aggregation. And when you do these measurements, that you basically have to add Sykes exclusion chromatography and then different continuous versus pulse flow techniques and everything. After 3 years, all these things worked great. And that's when we're at, nobody has ever done this proteins before. So for us, what was supposed to be, again, just a small one-off proof of concept for our protein, Lisa Ahlet meant, well, nobody has done this in any membrane protein before. There was one group that tried a slightly different technique. So Marie got a beautiful PNAS paper out of this, A, on the methodology, but then B, we were actually able to resolve differences between low and high pH for these channels. So here too, the differences are not large here, but in particular in this intermediate ranges that corresponds roughly to the type of size changes that we would see when these domains move in and out. We actually do see a clear difference in the measured data on the green curves here and the different models. So I won't go into detail about how we calculate the forward models here. The only problem is that we couldn't really use that to tell anything apart. That it appeared to be that the closed models were always the best fits. And that again made us a bit disappointed. Until we had that revelation in the other experiment, most channels should be closed. And then Marie came up with the idea, but just let's try the simplest, let's try linear combination. And that's when you realize the best, and this is not that such a strong result, but we actually get a slightly better fit if you mix in roughly up to 10 or 20% of open conformation, but allow 80% of the signal to come from closed channels. Now, if you just look at that curve is so small that we even put it in the appendix. It's not really hardly statistically significant. But then we have the advantage, we have a ton of different structures of these channels. We also have these 100 different Markov state model simulation, right? So we had a millisecond of simulation data. So first Marie started to do all these forward models, but not based on X-ray structures, but based on all our simulation data. And then we got a whole range of different curves here that actually overlap the experimental structures really nicely, either at high pH or at low pH. And there is a bit of a shift downwards in the channels here. So what we eventually then started doing that, instead of being stuck in this old-fashioned way of thinking that we're comparing one structure to one data, this is a measure in the experiment at the neutron source. This is a measurement on an ensemble of channels. So instead of just trying to pick one end state from a structure, we have 1 millisecond of simulation data, both at high and low pH. So what we should of course do is compare this to the entire MD ensemble. And let's see, I'm going to skip this in the interest of time. And the cool thing, so we're picking all these different end states from these 100 10 microsecond simulations, 100 different frames. And then we're comparing the last, I think it's 2 microseconds from each and see how well they fit. And the cool thing is that it's awesome. So if we do the crystal-based structures, they have fairly horrible fits. Then we energy minimize the model, we equilibrate them, and after say 200 nanoseconds, the last 200 nanoseconds from the equilibrate trajectories, both at high and low pH, fit better than anything else. And this is, I kind of feel this is the revival of MD a bit, right? I like MD, but we're always in this, when you're comparing to experiment, they don't fit that well. And then you have to argue your way around it, that there might be a reason, there might be shortcomings in the force fields. Yeah. We're basically looking for excuses. Yeah, those are the X-ray structures, those are our results. So I think that the recurring theme here is that I think most of our problems is really in sampling, not the quality of the models. And as long as we have good sampling and comparing our ensembles, they fit surprisingly well. So that we might just have been lucky here. We have other channels too. We have been able to show this for another channel. So in this case, this really confirmed our model of the clip gating. And I'm gonna need to rush through the last few slides here, but we have an entire new channel. And this is also something that we're just submitting to publication. This is a new channel called diclic from another bacterium, and that is known, it has an entire N-terminal domain, kind of 2 domains in the N-terminus here. So there's a much larger floppy part on top of the channel. And here too, we were able to, about a year or 2 ago, determine open and closed X-ray structures with microLGIC. And they're awesome because we have structures, but they're also horrible because the open X-ray structure, we can't get this to be stable in the simulation. They are so open that lipids can kind of move in and out here. And it is an X-ray structure, and Mark is one of the best X-ray crystallographers I know. This is obviously what we have in the structure, but I can't get this to be stable in the simulation. So what Marie has done, she has both done cryo-EM structures of this and small angled neutron scattering of everything, including these N-terminal domains. And the advantage here, now we have a channel with a much larger motion. So there's more of a difference between the open and closed states. And the cool thing is that we see this. We see the entire N-terminal domains that we can predict. We can't predict exactly how they move, but we can predict their up versus down state. And in particular, the SAS data here in the open state is not compatible with the X-ray structure. The SAS data claims the channel is not that open in the open state, which was fun. So that And that was also confirmed in our cryo-EM structures. So we actually have new high-resolution cryo-EM structures of the click, which we're submitting and about to submit to. And these are stable in simulations, in contrast to the X-ray crystallography data. We also think that we have a fairly good view of specifically what happens in the external domain. And again, this is an entire separate chapter I can talk about at some point, but we've identified binding site for calcium ions, and depending on whether we have these calcium ions bound, that's causing an entire shift in the flexibility of the extracellular domain here. That might, where we believe that this channel is actually gated by calcium ions, which is fun because we probably threw every single chemical we had in the lab, until we realized that the only thing that matters is how much calcium we had. And the one thing that we hadn't systematically varied turned out to be what was gating it. In the interest of time, I'm not going to go through the movie here, but to sum things up here in some sort of fashion, much of this work is still going on. We're doing much more experiments with years. For a few years, I think that we were about moving away from MD simulation more towards experiments. I don't think we are anymore. We have very much moved back, and I love the fact that, for instance, that MD simulations combined with these extremely low resolution, this is hardly a structural technique, right? You're having a one-dimensional curve. But the more we do that, MD, this is a match made in heaven, because MD gets you all the detail you want. You can put a flag on every single atom. The one thing you do not get from MD is an absolute answer, but that we get from the other techniques. So as long as MD provides you with a model, the other technique will tell you, is that model right or not? And that's in scope, it's kind of similar to electrophysiology, where we all started from. There are a ton of people here that have helped with various aspects of this. The one I would be completely lost in the lab without Rebecca. Stephanie did a bunch of the early studies in the lab together with Rebecca. Urska and Marie, there are our structural people. And then it's, well, almost half the people in the list here have helped with various simulations of these channels. And I tend to drag in half the GROMAX team when we need help with the simulations too. And then a bunch of experimental collaborators for the projects. And then the real heroes, the ones doing the work while I'm here enjoying myself. Thank you so much for your attention. I'm happy to take any questions.
Wonderful, wonderful talk. Thank you, Karas. So this was amazing. So you believe in what we think is the open structure experimentally?
Yes, I've gone GLIC. The former putative open GLIC structure, it's an open structure. It's really well known.
Okay.
It's not that one is not desensitized.
Okay. So then I guess part of the problem, going back to your question about inhibition versus stimulation by the same molecule, goes back to the mechanism of activation and desensitization, right? So do you think we understand the mechanism of what happens to the channel when it becomes active at the molecular level?
I would say that I'm fairly confident that the mechanism between the normal gating, the activation that happens with the upper part of the channel opening and closing. For most of these channels, I would say that we have enough data that I'm fairly convinced that the entire desensitization process is rather the lower part before collapsing.
I see.
Now, the really hard part of this is how much the opening, the activation happens with the extracellular domain. We're having magic happens in the extracellular domain, right? Something binds and causing these subunits to interact and twist a bit. And then there's a protein quake that's 60 angstroms over a protein. Something is pushing down. This is where you got to believe. There is a miracle happening here. And when that happens, and I think the desensitization process is really some sort of strength that we're getting to an intermediate state that's not really stable, and then we have to do desensitized one. What I have no idea about how we get back from desensitized to closed, because you don't have to open it. You go directly from desensitized to closed. So one way or another, this whole process kind of resets, but it's not going the same way back.
Absolutely.
We tried to address that with Markov states model, but this far it doesn't work.
Interesting. Questions? So I hope that you all heard the importance of sampling. Okay, Sam?
Oh, yeah, hi, can you guys hear me?
Yeah, loud and clear.
That was just a beautiful talk. But what I missed, unfortunately, did all my favorite ions except magnesium or zinc. And I know at least the channels that we worry about in our minimal cell simulation, they're ATP activated. And the little bit of regulation that's left in the cell seems to be done through a protein that binds to the ATPase domains. Have you looked at any of those types of problems where you have actually a protein helping with the gating and not just the gating by
No, I think you have no idea. We're super interested in it. The problem, we have no idea what other proteins should be. The one thing we do know is that the postsynaptic membrane is complicated. There have been some cryo-ET, so tomography studies of it, where you can start to see the entire distribution of GABA channels. But of course, that we don't have any resolution of a synapse with our prokaryotic channel for obvious reasons. They're not in any synapse. I think for these studies, I would suspect that this could be a significant difference between pro versus eukaryotic systems. So in a eukaryotic system, I would almost be a bit surprised if it's not interacting with other proteins and the postsynaptic membrane. But I have no idea what protein it is. If there are not half a dozen people listening in online here, together with a colleague, we're thinking of writing a grant and trying to go out and model the entire synapse, both the pre and the postsynaptic membrane and everything, and do that with electron tomography. And that might allow us to see interactions with these proteins, but then it's going to be super coarse, 1 or 2 nanometer resolution, right? But if you just start identifying things here, we might be able to see with super resolution microscopy. And if you can just find what those protein partners are, you should, at that point, I think you should be able to start doing docking or MD simulations. So that I think you're right. I think they are interacting. It's just I have absolutely no idea what the partner is.
Yeah.
And then could I ask one more question? You were almost got up to it when you're talking about lipids and mixtures and more realistic scenarios. I remember how fascinated I was when I saw the first simulation, I think is maybe Imad and Klaus had done with something with had a lot of cardiolipids in there. You could practically change the curvature of the membrane. So have any of your studies included, titrating not only the amount of cholesterol, but perhaps cardiolipids in there?
We not really titrating. We have prepared the problem. Titrating this is complicated because you can no longer do it in an oocyte, right? And an oocyte is it's a beautiful, simple, and easy system to work with. What you then need to do is that you need to create liposomes, which is possible, but it's like suddenly everything takes 100 times longer. And then we can measure currents and say, starting with the pure POPC membranes, and then you actually can titrate it. And that's how, for instance, that's how other groups have been able to show that even cholesterol presence has an influence on GLIC. We have simulations where we started to see that PG appears to be important for GLIC, and we appear to see significant differences between GLIC and another channel called ELIC. So we are kind of collaborating with Weilan Cheng and a few other groups. We think we have binding sites. So cardiolipin is an extreme example, of course, but if we can show this even for PG, I think it would be super cool. Also, what I didn't have time to show here because I didn't ask my students for time, that we actually obviously resolve lipids in simulations, right? But we now have new cryo. So we used all these Markov state model trajectories and tried to identify not only the open versus closed state, but can we see differential lipid binding? And also look at, can we see open versus closed? Are there differences in the lipid conformations we resolve? And we see that. The cool thing is that these match, I wouldn't say perfectly, but they match with differences we see in the open versus closed cryo states, where we now repost all that data. So we can see lipid density in our cryo structures. And there are at least a few cases where they overlap beautifully. That's it's perfect. They match both the head group and the tails and everything. And it comes back. I probably trust simulations more today than I did 10 years ago. It's kind of remarkable how useful they are.
So part of the activation is actually lipids moving around these channels, then, kind of.
I'm not sure. Well,
yeah.
I think it's the same type of lubrication, right? That you might need certain types of lipids to make it favorable enough for the channel to open, so that the lipids per se will not be able to open the channel.
Sure.
But you likely need a certain lipid composition to allow the channel to open.
Cool.
So that for eukaryotic systems, you know that most of these proteins most of these channels work. They hardly show any conductance unless you have cholesterol. And for 10 years, everyone was having different models where the cholesterol is binding. we still have hypotheses about that.
More
The
than
question
that.
hasn't been resolved.
Charlie,
Thank you.
Zan, are you done, Zan?
Oh, yeah. Oh, I could ask questions all
Okay.
day.
Okay, sure. So I don't want to move on without you being done. Charlie,
So you mentioned that only a few percentage of the channels become open. So evolutionarily, why do you think that's the case? So you can have less channels, but they are more efficient in becoming open, right?
It likely has to do with this desensitization process. So that, and again, this is why GLIC has some challenges that we don't have a formal agonist. But if you have, remember that if you're opening the channel, it's opening and then it's immediately shutting down again. And as if you now have an entire ensemble, and once you have shut down this desensitization, it takes a while for you to recycle before you can open again. And if you now have an entire ensemble, even if you keep this under activating condition, a large fraction of your channels are going to be in the cycle that they're now undergoing the recycling. That's the reason why we're a bit uncertain about this for GLIC, that to recommission, you need to do single channel recordings. That's where you can actually see the desensitization happen.
Until you have that.
Yeah, until you have that, exactly. And that's why, so in our own conductances, this is not based on my data. This is based on E-Link, and you've done it too. So if you do whole cell patch clamping, you can't get that conduction, and then you can't measuring this open probability specifically for a channel part, and you can't do it without single channel reports.
interesting.
So the channels are not all in the same phase. Some of them are
Recycling,
some of them
yeah.
are open, some of them are recycling.
So how many of these channels usually you have in a synapse? Tens of millions,
Many.
In a synapse,
Tens of thousands.
tens of thousands. So it's
gives enough
easy
signal
to,
be robust.
yeah, cool, very interesting.
And in our small cells, right, remember that
Yeah.
we've overexpressed this problem by at least 2 orders of magnitude. So 99% of the channels on the surface of the protein. So that for an oversight, that would be a significant.
Cool. Well, yeah, sure. Very cool. Other questions? So I have one last question. So for GLIC, it's proton activated, right? So for your simulation, are you protonating some stuff?
Yes.
Do you know where-
This is a you protonate. There's lots of PK calculated.
Right.
There is not one single residue.
Sure, that's
responsible
exactly
for
my
this
question. Yeah,
I
okay, okay.
marked it out. We had a student
Yeah.
go through. They mutated pretty much. They mutated every single titratable
Titratable
residue one at the time,
Yeah, okay,
and they
fair
couldn't find
enough.
it. So it's a collective effect.
Fair enough, fair enough. Okay. Okay, thank you very much, Erik. It was wonderful.
My
Let me,
pleasure.
beautiful, beautiful. Sampling, guys, sampling. Don't forget that.
Yeah. Hey, Iman? Iman, just a quick question to you.